effect potentiate once it was established for simply an hour. And so this experiment, which was done in the late 80s, early 90s, which we reproduced, basically said that most protein kinases are not important for the mating. So source-foreign affects CAMK2, affects most forms of PKC, protein kinase C, affects the cyclic AMP-dependent kinases, hundreds and so on. But as you'll see, the devil's in the details. And so what I was interested in was perhaps the very simplest way you could have a molecular memory from a protein kinase. And it was very simple because there was one particular family of protein kinases which had been implicated in short-term memory, but had this really simple way of becoming a persistently active kinase. And that's key. And that's called protein kinase C. So protein kinase C, shown here, has a catalytic domain in green and a regulatory domain in red. And the regulatory domain contains a pseudo-substrate, which is around 13 amino acids, that fits into the catalytic site and locks it down because it cannot itself be phosphorylated. Okay. And that's important for keeping the kinase in an off position. Now, protein kinase C is normally activated by second messengers, such as diacylglycerol, or for some isoforms calcium, that will bind to the regulatory domain and cause a shape change in the kinase, allowing access to a substrate that can get phosphorylated. But these second messengers are rapidly metabolized within seconds. And so the enzyme is going back and forth reversibly from this inactive to the active state. So it's like your keys on a keypad on your laptop. They have little springs in them that keep them in the off position. And that's absolutely essential for them to act as signal transduction devices. They're not information storage devices, like a hard disk. But there was a very simple way of turning PKC into an information storage device. And that was because when PKC was in its open conformation, it was now susceptible to limited proteolysis that removed the regulatory domain with its auto-inhibitory pseudosubstrate from an independent catalytic domain, which was called PKM form of PKC. And because this PKM was no longer being inhibited, it could now just continuously phosphorylate and act as a storage device. The problem is that this was always a test tube phenomenon. It had never been shown in a real cell that a PKM form of PKC got made. So when I started my lab, what I did is, my hypothesis was, well, maybe there are some isoforms of PKC that are activated by this translocation, but maybe there are others that make a PKM form during long-term potentiation. And so what I did is I made antibodies to all the known forms of PKC, of the three different classes, conventional, novel, and atypical. And I made them to the C terminus so that on a Western blot, where we can separate by size the different proteins and then recognize them with the antibodies, we could see either a full-length form or, if it existed, a shorter PKM form. And so what we found when we looked in the hippocampus was nearly every PKC was expressed in the hippocampus. But there was only one form, the zeta, one of the two atypical isoforms of PKC, that had that shorter, smaller molecular weight fragment. And then when we looked at what was happening during long-term potentiation, what we found was all of the PKCs got active right after that tetanus, but that activation only lasted for around 15 seconds. By a minute, they were all back in their inactive state. The only thing that was persistent was an increase in that PKM form of zeta. And that increase was very persistent, and it correlated with the amount of of the synaptic potentiation. But there was something that was a little strange. And that was, while we were seeing an increase in PKMzeta, we were not seeing a decrease in what we thought was the precursor to PKCzeta. In other words, if there was cleavage, if PKM was going up, the PKC should have gone down. But that's not what happened. So we figured, oh, well, there must be new synthesis of the PKCzeta precursor that replaced that which got cleaved to PKM. And so what we did was we added to the bath a protein synthesis inhibitor. And like others had found before, that blocked that late phase of LTP. But to our surprise, it also blocked the increase in PKMzeta. And it took us 10 years to figure out why it was the formation of a shorter, persistently active fragment of zeta was being made in a protein synthesis dependent way. And the answer is quick to just be summarized in this one slide. The trick, the essence of it is in the gene of PKCzeta. And the gene has a regulatory domain, axon shown here, and a catalytic domain here. And the regulatory domain, which normally makes the full-length enzyme, that promoter is shut down in the forebrain of mouse. Instead, an internal promoter here makes a messenger RNA that can translate only the catalytic domain of zeta. And that message, or RNA, is transported out into the dendrites of the neurons. But that message is translationally repressed. It cannot make new protein unless there is, during that 100 hertz test, an activation of the NMDA receptor, which allows calcium to come in and activates an entirely complex signal transduction cascade involving CANK2 and PI3 kinase, actin formation, but they're all focusing on new protein synthesis, which allows for the synthesis of PKMzeta. That's the only time in which this happens in the hippocampal slice. So then the question was, okay, if we have this mechanism for making a persistently active kinase, and it correlates with the maintenance of long-term potentiation, well, is it functionally important to the potentiation? Is it necessary and sufficient to cause the potentiation? Well, so for sufficiency, what we did is we recombinantly expressed and purified the PKMzeta. And we put it into a whole cell recording pipette, and then attached onto a single CA1 pyramidal cell in a hippocampal slice, as we're giving that every 15-second afferent tetanus, afferent test stimulation. So as the protein kinase comes out of the recording pipette, there's a massive potentiation of the synaptic response that's mediated by the AMPA receptor. And so there's this increase of about, on average, a doubling of the number of AMPA receptors in the synapse. We did a technique called non-stationary fluctuation analysis to analyze exactly how this is happening. And it's basically by doubling the number of the functional postsynaptic AMPA receptors. Okay, so PKMzeta by itself, without giving that tetanus, if you just simply put it in to the cell, it'll cause potentiation of the synapse. But what about that key experiment about reversing potentiation that's already been established? Well, here's that experiment I started with. That staurosporin blocks induction, but not maintenance. Well, as I said, the devil's in the details, because PKMzeta turns out to be one of those kinases that's not inhibited by staurosporin. But there are two known agents, only two, that can inhibit PKMzeta. One is a compound called shellavethrin. So what we did was we had now two stimulating electrodes in the slice, both of them we give test stimulation every 15 seconds. But to one of them, we gave that 100 hertz tetanus and caused that potentiation. The other one, we just left alone and kept as a control. And so we're measuring two sets of synapses within the same hippocampus slice. And then an hour later, we add to the batch shallow-Riethman. And shallow-Riethman did what nothing else in the thousands of papers on an LTP have ever been shown to do, which is to reverse potentiation an hour after it has been established without affecting basal synaptic transmission. So we wanted, shallow-Riethman is actually a plant toxin. So we wanted something a bit more rationally based than something from a plant. So what we did is we took the pseudo-substrate region, those 13 amino acids, and then meristylated them so it can now, these peptides can now penetrate into the slice. And that was a very potent inhibitor of PKMzeta. We call them zeta inhibitory peptide, or ZIP. And ZIP did what shallow-Riethman did, but nothing else could do, which is to reverse the potentiation of LTP. In fact, we determined the IC50 of ZIP on PKMzeta's ability to act on the AMPA receptor in the synapse itself. We could do that because we could inject the PKMzeta and then add to the bath various concentrations of ZIP. And the IC50 for ZIP on the ability to block PKMzeta's ability to potentiate just by itself was nearly identical to the ability of ZIP to reverse potentiation even now 5 hours after the tetanus was given. Like, totally unprecedented. So, ZIP and shallow-Riethman and dominant negative, which we can also perfuse into the neuron, we all showed that the three known ways of inhibiting PKMzeta, they can all either reverse or block the maintenance of LTP. Well, what about blocking the translation of PKMzeta from its messenger RNA? What about knocking out the gene of PKMzeta in the nucleus? So if one blocks the translation of PKMzeta by having an antisense oligodeoxynucleotide that complements the start site of the translation, what we can show is we can prevent the formation of PKMzeta during LTP. And here's a controlled, scrambled version of the peptide, of the oligodeoxynucleotide that doesn't affect that. We can inject it into the animal, shown here as red, and we can do it on one side of the brain, into the hippocampus. We do the antisense that will block the synthesis of PKMzeta. Then the other side of the brain, the same animal, we can inject a scrambled version, which doesn't. And then when we do LTP and then break the code of what we injected, we find that the antisense prevents the late phase of LTP while the scrambled would not. But what about the knockout? Well, recently there were two papers that had reported that conventional knockouts of the PKC-zeta gene actually had no effect on learning and memory or LTP. So, well, how is that possible? Well, to really understand that, we have to understand more about the evolution of the gene of PKC-zeta within the context of the PKC family as a whole. So the PKC family as a whole split between an atypical PKC and the other two, which eventually became conventional and novel classes, around 800 million years ago in single-cell eukaryotes called Capsa-spora, which was sequenced a couple of years ago. So that atypical PKC remained as a single form in C. elegans, in Drosophila, and in Aplysia. But with the early vertebrates in the Cambrian period, around 500 million years ago, there was a large gene duplication of much of the genome of animals. And that single atypical PKC split duplicated into two forms. One was called iota, and the other was PKMzeta, which we've been talking about. But then immediately upon that split, in the very earliest record that we have at a genomic level, there was that internal promoter that I mentioned that made in the nervous system PKMzeta. So right away, as soon as zeta was made, there was a dedication in the nervous system to making that PKM form. But what I showed here is that even before that split, there's evidence that Aplysia and Drosophila use this atypical PKC in long-term memory. So what we found was we looked in detail at that other isoform, iota lambda. And what we see is that when we, in the conventional knockout, when there's no PKM zeta, there's an increase in what's normally only a trace amount of a PKM of the other isoform. And if you inject that other isoform, iota, which has 93% homology with zeta in its catalytic domain, which is very, very high, it can also potentiate synaptic transmission. So there's basically a backup plan in mammals for the formation of a PKM form of the other atypical PKC when zeta is conventionally knocked out. But if you do a conditional knockout and eliminate the PKMzeta in an adult animal, the message level is shown to drop, the increase in PKMzeta and LTP is blocked, and there's no late phase LTP. Okay. So blocking PKMzeta at this level of the gene, or translation at the level of the message, or at its activity in terms of its unique persistent activity in terms of the gene product, which is the enzyme, they'll all affect long-term memory. But the unique thing is that blocking that activity can reverse LTP that's already been established. And so what that means is that for the first time we had a tool with these PKMzeta kinase inhibitors. We had a tool for asking the question, is the maintenance of LTP, does it have anything to do with the storage of long-term memory? So to make those baby steps from the hippocampal slice, physiology, into behavior of awake, behaving animals, we had to figure out, well, what's the right amount of this compound Z which will reverse LTP in the hippocampus if you want it to stay in the hippocampus. And so what we did is we did in vivo LTP and then injected as a bolus the ZIP. And that we found the right dose that reversed LTP, that was able to reverse the LTP even 2 millimeters away because it's thought that the memories in the hippocampus are sort of widely distributed in animals. And again, just like in the slice, the ZIP had no effect on baseline synaptic transmission. Okay, so now we had the tool, and we knew how to use the tool to be able to reverse LTP around the day after it had been established. So what we then needed was a behavioral task that was of something very similar to an LTP experiment. Well, that's kind of difficult. If you do like a water maze, it can take days for it to learn anything. All right. So if you want to stay with a spatial task, there's really only one very rapidly learned spatial task. It's a little unusual. It's called active place avoidance. But it had a lot of advantages. It could be rapidly learned within around 2 hours, and that memory can last a day. Repeat that, the memory can last a month in the hippocampus. So what it is you put an animal, a rat or a mouse, into this rotating carousel. It has clear plastic walls. But then there's a non-rotating pizza wedge-shaped sector right here. And the only way that the animal can avoid getting that shock is not to pay attention to the information that's on the floor. That won't help. It has to pay attention to the cues in the room. And that makes it a very spatial task that really engages and requires the hippocampus for both short-term memory and long-term memory, and even remote memory for a month. So looking down now with the camera, this is what you see. So if you put a rat in the apparatus with the shock off, the rat tends to stay on the outside, maybe looking through the clear plastic wall. And so the training is to turn the shock on in this area here for 10 minutes. And then take the rat out and put it in its home cage. And then repeat that 8 times. And by the end of the 8th training trial, the rat has figured out to avoid this area here. Now, the information, as I said, that's on the floor is useless to it. It has to ignore that and pay attention to the cues in the room. So we waited, trained the animal, waited a day, just as in our LTP experiments, and then injected saline as a control into both hippocampi. And we put the animal back into the apparatus with the shock off. Those animals remembered to avoid. Now the memory's not perfect, but it didn't get a shock there. Then we take another set of animals, train them, wait around a day, and inject zip into both hippocampi. And put it back in, with the shock off. No evidence of the retention of spatial memory. So you can track this kind of movement of the animal over a 5 or 10 minute period by this kind of track here. And you can see that a control animal, they remember, they avoid, and then a day later they still avoid. These other animals, they avoid, and a day later they're injected with zip, and 2 hours later, just as in our LTP experiments, they're put in. And if you reverse LTP by inhibiting PKMzeta, they have no evidence of memory retention. Long-term memory retention. Short-term memory, which also requires the hippocampus, was totally unaffected. Antisense, I'll just quickly show you, antisense now injected bilaterally will block the increase in PKMzeta that you can actually see after memory training. We'll talk about that at the end. And that completely blocks the retention of the memory. You take a conditional knockout and inject, in this case, we're injecting a virus that expresses free recombinase bilaterally, so we're knocking out the gene locally in the hippocampus, and that had exactly the same effect. So now, what's actually happening in the hippocampus to the neurons when we're erasing their memory? So we can actually study that because at Downstate, there's a large group of scientists who work on place cells. Now, place cells are basically pyramidal cells in CA1 or CA3. And if you put a rat or a mouse into now an arena environment, in this case it's a box, and you record from single neurons, pyramidal cells, what you'll see is the rat is sort of walking in like one particular area of the box, that particular neuron fires at a very high rate. A second neuron will fire at a high rate, shown by these colors, in another area of the box. And so it's thought that the whole ensemble of these different neurons, each firing at a different location, is what they call a cognitive map of this particular environment. Again, there are lots of pictures on the walls. So here's a control experiment. We basically put the whole hippocampus asleep. We did that with a drug called mucimol that activates the GABA-A receptor. And after the drug has been injected, the whole hippocampus is silent. The animal's still walking around. Now, the next day, we put that animal back into the apparatus. The drug is worn off. But what is the firing pattern of these place cells? Well, they're extremely similar to what they were the day before. So you don't need the continual activity of the hippocampus for the stability of the memory of the plate cell. Well, what about zip? Okay, here's another rat, and here are some more plate cells. This control, we inject saline, and that did nothing to the location of the plate cell. But then we injected zip that erased memory, that reverses LTP, and inhibits PKMzeta. And what happens to the plate cells is they kind of like melt, like hot wax. And then as the drug begins to wear off, and you put the animal back into this box, they reform. And initially, when the drug is still there, they're like forming and collapsing and forming and collapsing, and eventually they stabilize. But the location of these plate cells has no relationship to where the original plate cell was. So you basically erase this mass. Now, since no one had ever erased the memory before, and it was kind of heretical in the long-term memory field, it was, lots of people did lots of different types of behavioral experiments in which they're injecting PKMzeta inhibitors. And I won't go through all of them, but the point is that either in the cortex or in the amygdala or in the striatum, even in, as I said, even in the plesia, which doesn't even have a brain, all these memories, or rather, sorry, most of these memories, but not quite all of them, were able to be erased by inhibiting PKMzeta, or in the case of diplegia, in the homologue of PKMzeta, which is the atypical PKC found in diplegia. And so here's some of the properties. The same three compounds that can block LTP maintenance also disrupt established memory. Now the zip, which is a peptide, it has a half-life of around 2 hours. But it can disrupt memories that are 1 day old, even 3 months old. You don't have to be retrieving the memory at the time that you're injecting the zip. So it's not like another group of experiments called reconsolidation blockade, in which retrieval is necessary. Most experiments showed no spontaneous recovery of the memory for at least 1 month. But importantly, after the drug washout, the subsequent relearning and memory storage is normal. In fact, you could erase the memory and then teach the animal all over again, make a new memory, and then erase it a second time by injecting the zip in the same place. And that's important because the brain can be flexible. Maybe it's making that same memory in another region. And then the mechanism by which zip erases LTP maintenance and long-term memory is through the disruption of the AMPA receptor traffic. Okay. So let me show you an example of one of the longest memories that's ever been erased. It's another type of memory that humans have, as well as other animals. It's called, very simple, it's called conditioned taste aversion. And the advantage of conditioned taste aversion is that it's simple, it's very robust, it's very long-lasting, and it's also in the neocortex, which is thought to be where most long-term memories are ultimately stored. So if you give an animal a novel taste, say saccharin in water, and then within an hour or so inject a substance such as lithium that can make the animal sick. And then hours, if not weeks to months to years later, you give an animal a choice between the saccharin-laced water or regular water, they'll avoid the saccharin-laced water. And you can just measure this with a simple aversion index. And this memory is thought to be encoded and stored in the insular cortex, which is where the taste cortex resides and where the visceral malaise afferents are coming together. And so, if one does very strong training by repeating it, you can have a memory that lasts 3 months. And then on day 95, you inject zip. As I said, the zip lasts only around 2 hours. And then test the next couple of days. What we see is that the memory that's even many months old is disrupted. So now, another question is, well, if we can erase a memory by inhibiting PKMzeta, maybe we could enhance an old memory. Now, there are different ways of enhancing memory, but they're all involved in enhancing that initial consolidation of memory. That's just like for half an hour or an hour after you learn something. No one had ever been able to enhance a memory that's a week old, for example. So the approach that we took is with lentivirus. So we can inject lentiviruses that express PKM in the insular cortex. So we first reproduced the results with the zip by injecting a dominant negative mutated form of PKMzeta. So here we do the conditioned taste aversion, wait around a week, inject the virus, wait around another week for it to express the PKMzeta dominant negative. And just like in zip, the memory is less. So now to ask whether we can enhance a memory, we don't want to give such a strong memory, we want to give a rather weak memory. So we simply give less lithium chloride, the substance that makes the animal sick. That gives a less robust memory. So first we injected the virus that makes PKMzeta, waited for it to express, and then did the training. And then we looked 2 days later. And with the PKMzeta around during the training, there was a robust enhancement of the memory. But the unprecedented experiment was this. If we do the training first, and then inject the virus, and then wait around a week, and then test, even injecting PKMzeta a week after the memory had been consolidated was able to boost up the memory. So, if we can now uniquely erase a memory by inhibiting PKMzeta, and uniquely enhance an old consolidated memory by adding more of the enzymatic activity of PKMzeta, what about actually visualizing a memory trace if PKMzeta is a candidate molecule for the memory trace? So here's this active place avoidance. So what we're doing now is we're looking now a day after a rat has been put on that carousel and trained. And we look what's happening to the PKMzeta within the hippocampus. So here's a control animal that was put on the apparatus without getting the training. We still see this PKMzeta shown by this immunocytochemistry. Here's the hippocampus, here's the neocortex, here's the thalamus. A day after the training, we see there's a big increase in PKMzeta, particularly in the CA1 region, particularly in this layer here, which is where the input, the perforant path input of the entorhinal cortex is coming into the hippocampus. Other parts, like the dentate, is rather quiet. Thalamus doesn't change. The neocortex also increases. So it seems that there are molecular traces of memory a day after the training, not only in the hippocampus, but also in the neocortex. We can make sure that our antibody is specific by doing that conventional knockout and showing that here's the control. This is a close-up of the CA1 region of the hippocampus. Here's the control, here's after training, and there's a knockout. So our antibody is just detecting PKMzeta and nothing else. Now, as I mentioned, the PKMzeta zip experiments show that you need the persistent activity of PKMzeta even a month after the training. So, is there a memory trace a month after the training? Well, it appears that there is. So, again, here's a control animal, and here's a month after training, and you can see that PKMzeta is still pretty much in the same place. Increases in CA1 in that perforant path, and in the neocortex. So now you may be thinking, how is this possible? How can you actually see a memory trace from a single memory? It just doesn't make any sense. The whole brain would just fill up with PKMzeta within a couple of days. Well, there's an assumption there. The assumption is is that PKMzeta is in every synapse. And it turns out that that's not quite right, because it's actually in a very unusual distribution. So we're now looking at an untrained animal, and we're injecting the CA1 pyramidal cell with a dye. And that dye will fill out the entire dendrite and fill up all the spines. And then we can do immunocytochemistry together with that to see how many spines, which is where the excitatory synaptic connections are going to, actually have PKMzeta in a resting state. Well, it turns out, even though this looks like there's a lot of PKMzeta there, in fact, it's only in around 1 out of 200 spines. But when it's in that spine, electron microscopy shows that there's a lot of PKMzeta in clusters underneath this postsynaptic density, or within the postsynaptic density, or right next to it. So I think that's why you can have lots of different memories, because the amount of PKMzeta is actually in very, very few spines. There's also another feature which I'll just mention, is that we've talked about LTP synthesizing PKMzeta, but there's another type of synaptic plasticity called long-term depression. And long-term depression is the only thing that we know that degrades PKMzeta very rapidly. So we think that what's happening in natural memory situations is like there is a combination of PKMzeta that gets synthesized and some PKMzeta that gets degraded. And it's that pattern of PKMzeta that's in these spines that's equivalent, the analog, so to speak, from the zeros and ones that's in a hard disk. And so I'll just conclude that since our studies of PKMzeta, there have been many, many other labs that have been looking at this kinase in disease state. And actually most interesting, where people seem to find, probably my bet is where it's going to come to clinical relevance the soonest, is in central neuropathic pain. Because in the anterior cingulate, but particularly in the spinal cord, lots of, what in animal models, it's very consistent, many labs have shown that the persistence of pain that is caused in chronic neuropathic pain, it seems to be related to PKMzeta-mediated LTP in the pain pathways in the spinal cord. So there, we don't mind erasing all of the memories in the spinal cord. There probably aren't that many memories in the spinal cord. Plus, you can always relearn them. Okay, so it's not like in the brain. Other people have found that addiction seems to be mediated by PKMzeta, post-traumatic stress disorder, and even there's experiments on Alzheimer's disease in which PKMzeta, and I must now add the iota form, which is that backup plan that I mentioned, that both, those are the two isoforms that form aggregates in neurons that also have aggregates of tau. And so, in conclusion, PKMzeta is a persistently active isoform of atypical PKC. It's synthesized during LTP and learning. That persistent activity of the atypical PKC maintains late LTP. And that persistent activity maintains many forms of long-term memory, including spatial, also trace memories in hippocampus, fear-motivated memories in the amygdala, didn't have time to show that, or the reward-motivated memory in nucleus accumbens, habit memory in the striatum, and elementary associatives such as acquisition taste aversion, and skilled motor memories in the neocortex. And we think that the PKMzeta is maintained through both its persistent autonomous enzymatic activity and the fact that the protein under some conditions seems just very, very stable. There may also be positive feedback loops of PKMzeta regulating its own local translation by its dendritic messenger RNA. And therefore we conclude that PKMzeta is the first identified component of the molecular mechanism of long-term memory storage. That backup plan, PKM-iota, compensates in the constitutive knockout, and that may also play an important role in memory maintenance. And so I want to thank my many collaborators, thank NIH and other support, and thank you guys, Berkeley, for inviting me here. And I'll be happy to take questions at the end of the meeting.